Hello everybody, my name is Mr. Christopher, and I'll be with you today to talk about Ester Synthesis. Now, in the laboratory we were able to take a putrid smell and change it into something that smelled sweet. An analogy I usually think of with what we did in the lab was we took something that might have smelled bad and made it into something sweet. How could you do that? What would be an example of this in real life? Well, sometimes when you get sick, you vomit. Vomit has a pretty bad smell. Now imagine you could take that smell and turn it into something that smelled like bananas. Would that be a good thing to do? Imagine if you had something that you thought smelled bad to you, could you change it to something that's good, or different, or sweet? So we're going to look at that today in what's called analyzing ester synthesis. So in the lab today, we looked at what happened in the experiment. What happened to cause the acid molecule and an alcohol molecule to come together and form a sweet molecule. So what was going on at the molecular level, at the atomic level, to change this smell? We're going to also be able to define a chemical reaction and review that concept and define what's called a catalyst. In this activity, we used a catalyst to help speed up that reaction. The first thing we know is chemical equations. A little bit of review. It's a sentence that keeps track of what happens during change in matter. Chemical equations are written with chemical formulas and keep track of the atoms involved in the changes. We know that the mass before and the mass after the reaction has to be the same with the number of atoms. In the test tubes here, you may have gotten different smells than this, but these are some samples of smells you could have gotten. For example, we had acetic acid isopentanol, started out putrid, then went to a fruity banana smell. Acetic acid and butanol was very putrid to begin with, and it gets a fruity pear smell. Butyric acid and ethanol, very putrid with butyric acid and ethanol, and has a fruity pineapple smell when you finish it. And then the number 4, the salicylic acid and methanol, is the smell you see in wintergreen lifesavers. It's a molecule that's made when these two are added together. And we're going to learn the name of that molecule and analyze what happened. So to analyze what happened, we need to know, first of all, the products smell sweet. If they smell sweet, we can assume that they have a certain functional group. In this case, they all contain the ester functional group. Many different acids and alcohols can be brought together to form an ester and water. So you can take multiple different ones. We only did 4 different combinations. But you could mix and match to form different esters. Now, in an esterification process, which is this reaction, this acetic acid and this butanol are added together. Now, if they're added into the test tube, nothing happens. Nothing occurs. The smell is bad. What we had to do was we had to start the reaction. To do this, we used a catalyst and we used energy in the form of heat. We heated it up. That heat allowed the molecules, acetic acid and butanol, to move faster. These were moving around faster, so they're more likely to collide with one another. When they collided with one another, again, the catalyst helps this process occur easier, and they collide together and they break at roughly these positions. Now, it could be that all of the atoms break apart. It could be that some of them break apart. But the simplest explanation here is that the acetic acid breaks between the C and the O, the butanol breaks between the O and the H, and this C attaches to this O to make butyl acetate, and what's left over makes HOH, or water. This is an esterification reaction. In this chemical reaction, the reactants were the things we started with at the beginning of the chemical reaction. They're written to the left of the arrow. Products were the things that were written to the right, and they, again, in this case, was the ester and water. Catalyst is the one thing that we looked at today, and the catalyst in this case is a substance that accelerates a chemical reaction, but is itself not permanently consumed or altered by the reaction. It is written above the arrow in a chemical reaction. So that means it's not a product, and it's not a reactant. Again, it's just something that is used to allow the reaction to occur quicker, and it accelerates this chemical reaction. When the atoms are rearranged during chemical reactions, not all of the bonds must break. Okay, they all could break, but a lot of them would form back together. And so the simplest example is when they break apart in one location. Now the name of these chemical compounds, these esters, is not random. For example, it says formic acid reacts with octanol to form octyl formate. So you can see that the octanol, the alcohol name, comes first and is written as an octyl instead of octanol. And then the formic changes to a formate because of the presence of the ester bond. This octyl formate is an ester molecule. Formic acid is a carboxylic acid. Octanol is an alcohol. Octyl formate is an ester molecule. Now, when we took in test tube number 4, this is not in your lab manual. In lesson 8 in your lab manual, okay, you're going to be completing lesson 8 and drawing what happens with these structures. This molecule here, salicylic acid, when it was added to methanol, you can notice that at one of these locations is where the bond needs to break. You've got all of these OHs here. So you can see here that if it attaches here to this O right here, I think I did it that one there, it attaches at the end where the double bond is. So the salicylic acid, this is the carboxylic group, so that's where this attaches here. The other OH just stays in place, the alcohol functional group here. So this is called a carboxylic acid functional group, and this is an alcohol functional group. This one has an alcohol functional group as well, and these cannot bond with each other, so that's why the alcohol and the salicylic acid come together to make this molecule here, which is known as methyl, because the alcohol name comes first, methyl, and then this goes from salicylic acid to salicylate, methyl salicylate. And this is the ingredient that is responsible for the wintergreen smell that you noticed in the laboratory. Now, this is an esterification reaction. This is all you have to know to put it together. Make sure you don't forget about the water molecule. Water molecule is always a product in this reaction, and your catalyst is written above the arrow. So this is how to write a chemical equation for each of the reactions you did in the laboratory, which is one of the things we'll do on our lab report. Now, how do you name hydrocarbons? We've talked about naming them and that there's patterns to them. If you turn to page S79, which is right after lesson number 9, we're going to look at naming of hydrocarbons. So let's look at naming of hydrocarbons. To name a hydrocarbon, there are 3 things that are included in the name. The first thing is you always count the number of carbons. Okay? So, as you can see on page S79, there's a chart that gives the prefixes here. Okay? 1 is meth, 2 is eth, 3 is prop, 4 is bute, 5 is pent, 6 is hex, or hexa, but in this case it's hex, 7 is hept, 8 is oct, 9 is non, 10 is dec, and those are the prefixes used initially for the name. So the first thing you do is fill in the number of carbons. Okay? Then the next thing you look at is the bond type. What type of bonds are located in this hydrocarbon? Now remember, a hydrocarbon is only made of hydrogens and carbons. So a hydrocarbon is only made of hydrogens and carbons only. So if they all have single bonds, it ends in ane. Double bonds, it ends in ene. Triple bonds, it ends in ine. So what's a good example of this? So let's say we have a molecule here. So here we have a molecule, and in this molecule here, let's see what we got. There we go. This molecule here, all of the H's are attached here to this carbon chain. So it must fulfill the honk 1, 2, 3, 4 rule and the octet rule. And when we look at this here, we should count the number of carbons first. 1, 2, 3, 4, 5 carbons. So it's got 5 carbons, so its first thing we write down is it is pent. Okay? Then we look, is it all single bonds? No, there's a double bond there. That double bond means it ends in an ene. And the last thing we have to consider, though, is where is the double bond? You need to be specific. It could be there, it could be here, it could be in different parts of the molecule. So what we do is we always want to find out the location of the bond. And we always count from the shortest end. So the shortest end here, the one closest to the double bond, that's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So this pentene is on the second carbon, so it's written in front with a dash. 2-pentene is that molecule there. So you look at the location of the bond, that's where the number comes in, and then you always count from the shortest end. So, if you look here at the examples on your page, there is a typo in the first example, so here's the correction to that. I'd like you to write the formula, the name for this formula. Then in example number 2, I'd like you to draw the structural formula for the names that are given. So nomenclature of hydrocarbons, it's got 3 main parts. Okay, you can do these, and we'll check those tomorrow. We'll see you. Have a good day, and I hope you come back real soon. Bye-bye.